Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about isolation of plasma membrane. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So basically we will talk about two techniques. Technique 1 is sequential centrifugation. So first you need to take some cells, and those cells are first broken open by mechanical shearing, that is by disrupting using a homogenizer. So you can take a homogenizer or mortar pestle that will help you to break down cells, and the cells are then mixed in the homogenizing solution. So now you are getting the homogenate. The homogenate is then subjected to a series of sequential centrifugations at increasing centrifugal forces. So we will see how they work. First, low centrifugal force for a short time. So here you have the homogenate. Now you have to centrifuge it at low centrifugal force for a short time. After the centrifugation process, the largest cellular organelles such as nucleus and then mitochondria and any remaining whole cells are sedimented into the pellet. So we all know that after centrifugation we get two products. One is pellet and the other is supernatant. So in the pellet you will get nucleus, mitochondria and other remaining whole cells. Now you have the soup here, that is supernatant. So you can transfer the soup or supernatant into a centrifuge tube, into a new centrifuge tube. Now you have to centrifuge it again. So after the second centrifugation process, the pellet will contain the plasma membrane. So what you need to do now? So you resuspend the pellet in 40% sucrose solution. Okay. Now prepare centrifuge tube with a 4-step sucrose gradient, that is sucrose showing increasing densities from top to bottom. So you have one more centrifuge tube here, and here you have kept the sucrose gradient solution, and the gradient is like that. Here you will get the highest density sucrose and here you will get the lowest density sucrose. In this way, that means from top to bottom the density of sucrose is getting increased. Now you will add your resuspended pellet in this sucrose solution. So resuspended pellet is placed at top, and then what to do? Next you will centrifuge it again. So here you have your pellet plus 40% sucrose solution that you have received from this centrifugation process, and now you are adding it in this sucrose gradient centrifuge tube. Next again you have to centrifuge it. So after the centrifugation process, the plasma membrane will settle down. So after the centrifugation process, the plasma membrane will settle down when its density matches with that of the medium, that is 40% sucrose. So here you have the 40% sucrose and here you will get the plasma membrane. Why? Since you have mixed this pellet with 40% sucrose solution. That's why you will get your plasma membrane along with the 40% sucrose in this place where you have 40% sucrose gradient. This is first technique. Next is technique 2. This is quite simple process. This is called adhesion method. So this is the simplest method. It involves the use of cationic polylysine, which can increase cell adherence by interacting with negatively charged cells. So polylysine, so lysine, we know lysine is the positively charged amino acid, and cell membrane is negatively charged. Why? Since cell membrane is made up of phospholipids and phosphate group carries negative charge. So there will be an interaction between the positively charged lysine and the negatively charged phosphate groups of the cell membrane. So what you need to do? First, take the petri dish where the plate is covered by polylysine. So here is the polylysine coated glass plates. Now cells are attached here. So you keep cells here. First you keep cells here. Now cells are attached to polylysine coated glass plates and then ruptured by hypotonic distilled water. So after keeping the cells in this polylysine coated glass plate, you just add hypotonic distilled water, and that is ice cold. Hypotonic solution will help the cell to get ruptured. Now plasma membrane remains attached to the plates after washing away the intracellular organelles. So this is the lysis method. After adding hypotonic solution in the cells will be ruptured, cell lysis will take place, and then you wash them. So after the washing process, all the intracellular organelles will be washed away and only the plasma membrane will remain attached to this polylysine coated plate. Why? Since there is the strong interaction between positively charged lysine and negatively charged plasma membrane. So this is the second way, and this is most simple way you can do to isolate your plasma membrane from the cells. So this is all about today's lecture. I hope you liked the lecture. Thank you for watching my video.